This protocol begins by adding incubation buffer, FITC-casein solution, and sample containing the protease of interest to a microcentrifuge tube, and then gently mixing and incubating at 37 degrees Celsius for 60 minutes in the dark. To stop the proteolytic reaction at the end of the incubation period, add TCA solution, gently mix, and incubate at 37 degrees Celsius for 30 minutes, again in the dark. This step will cause the unreacted substrate to precipitate. Centrifuge the sample for 10 minutes at 10,000 Gs. Remove the supernatant, which contains the acid-soluble FITC-labeled fragments. Transfer the supernatant to a cuvette or multiple well plate, dilute with assay buffer, and read the sample on a fluorometer.
Hi, I'm Carrie Cupp-Enyard, and I'm a scientist at our Sigma-Aldrich Dekalb facility in St. Louis, Missouri. Today I will be showing you a procedure for fluorometric protease activity determination. We use this procedure in our quality control lab to determine low protease activity. So let's get started.
For this procedure, the incubation buffer, assay buffer, and 0.6 normal trichloroacetic acid, or TCA solution, are provided as ready-to-use solutions. Aliquot the solution of FITC-labeled casein into smaller volumes and store at -20 degrees Celsius, making sure to protect from light by using amber vials or wrapping clear tubes in foil. Aliquoting the FITC-casein substrate minimizes the need for repeated freeze-thaw cycles.